Welcome back to our NPTEL course on scanning electron ion probe microscopy in material characterization. In last few class we have been discussing on scanning ion microscopy. In that topic we were discussing of different contrast mechanism. Today we will continue with that, and also we will move forward to different imaging modes. In the last lecture we have discussed on the topographic contrast, material contrast, ion channeling contrast. Three different contrast we have discussed in our last lecture. As we have seen that topographic contrast is much significant in helium ion microscope as compared to scanning electron microscope. It is because of higher secondary yield as a function of the tilt angle. When we go for different tilt angle of the specimen in degree versus secondary electron yield, then we have seen that for a helium ion microscope or ion microscope it goes like this. On the other hand, for a electron microscope it goes like this. The secondary electron yield drastically increases for ion microscope. This is for helium ion microscope, this is for SEM, because the yield of the secondary electron is higher with ion microscope at a particular tilt angle. Therefore, we have a better contrast in helium ion microscope. In the this is about topographic contrast, mostly for 3-dimensional specimen we always have sample up and down, and therefore, they have different tilt angle, and at different tilt angle we have different yield of secondary, we have different secondary electron yield. Second contrast is the material composition contrast. We have also seen that materials with different atomic number produce different numbers of secondary yield, and therefore, one can distinguish in one image whether there are two different kind of materials is present. Similarly, ion channeling contrast, which is almost similar as that of scanning electron microscope, when material is amorphous or crystalline, depending upon the orientation, the channeling contrast occurs. Voltage contrast, magnetic contrast, optical contrast are all similar type as that of scanning electron microscope. Therefore, I will not discuss those we have covered in our scanning electron microscopy part. In the scanning electron microscopy part, we say it is a cathode illuminations because our electron bombardment was occurring, but here in helium ion microscope we say it is ionolominescence because helium ions incidents on the specimen surface. What additionally I will talk here is ion dependent contrast. Ion dependent contrast is because we have different, we can use different type of ions in ion microscopy. All electrons are same, but not all ions are same. Therefore, we can change the ions and see what different contrast is being observed. And here is the study, recent study on nitrogen ion microscopy and helium ion microscopy of same sample. As you see in the left image here, in the left side image, we have different type of specimen, different type of materials, that are viewed under microscope in a nitrogen ion microscope. So, for example, here it is gold, here is gold, in this place is a collapsed gold. We have a gold, we have a graphite, we have SiO2, and we have also 4 layer graphene. If we carefully look, this is a helium ion microscope image, it is a nitrogen ion microscope image, not helium ion microscope. The first two one are the nitrogen ion microscopic image, and second two one these are helium ion microscope image, and they are taken at different ion dose. We could see in the leftmost image, this image, there is a contrast difference of different type of sample, gold, graphite, SiO2 and 4 layer graphene. For example, here in the graphene and graphite appears more darker, graphene and graphite appears more darker, and it is more clear when dose has been increased to an extent. You can see that graphene place, in this place it is completely dark. Similarly, graphite is also completely dark. Gold contrast is not same as SiO2 contrast. So, all these like graphite and graphene have similar contrast, on the other gold and SiO2 have a different contrast in a nitrogen ion microscopy NIM. On the other hand, if we see in the helium ion microscopy case HIM, then we do not see a different contrast with graphene. You see almost graphene contrast, graphite and SiO2 are similar. Certainly, gold is different, gold have a different contrast. One can easily distinguish gold, this is also gold, this is a pattern sample, this is also gold, indicating from their contrast we can differentiate there are two different materials, like here is gold and here is something else. Similarly, with a little higher dose we can also see the contrast difference, here it is gold and here is similar, all similar. We can only distinguish between two different type of contrast, one is for gold and another is for all other materials. But if we look at the nitrogen ion microscopy image, we have clear cut difference with from the graphene and graphite. There is almost negligible or no secondary electron yield from this region, graphite and graphene, no secondary electron yield. There is a suppression of secondary electron yields when we do with a nitrogen ion microscopy, but in this cases it is not. So, this contrast difference is the effect of ions. In case of ion, we could able to see the difference, but not here. That means, different ions interact with this specimen different ways and therefore, providing different contrast mechanism, which is not possible with a scanning electron microscope or electron microscopy image. If we look at little bit at a higher magnification, we zoom in and we can see here, the helium ion microscope dose is increased, here it was 10 to the 13, 7.7 and it was 8.3. When the dose was increased, now now we can see some contrast difference here from graphene and SiO2. Here is graphene, this is a zoom in portion. Here we can see clear difference, graphene, gold, SiO2, all difference we can see. By increasing the ion dose in helium ion microscope, we would able to differentiate to an extent about the presence of two different material, graphene and gold, graphene and SiO2 or graphite. Graphite is not seen here because it is a magnified image, but otherwise at a little lower dose we were not able to distinguish. But moreover, the contrast difference as you see here, the contrast difference in this case is significantly different with different ions. This indicates the importance of the ion microscope by varying different ions. Moreover, helium being the small size, smaller the size, having higher resolution, could able to distinguish the steps at this place, which is not as clear in case of neon ion microscope, because the nitrogen ion microscope, because nitrogen size is bigger, therefore resolution will be not as big as, not as good as of helium. So, the topographic contrast here, this is the topographic contrast is more significant in helium ion microscope as compared to the nitrogen ion microscope. Here is the person, the same person, but at a lower, much lower magnification, looking at the wider view, and this is the region we were previously looking at it in the previous slides. But what we can see clearly here, this is the region we are looking at it by helium ion microscope, here by nitrogen ion microscope. In the nitrogen ion microscope, the region we are looking previously, that region becomes completely different contrast. It is because those region have some interaction between the nitrogen ions and the specimen, thereby change in their change in their so there is a interaction between the ions bombarding on the specimen, therefore, the change in the structural features inside the material, therefore, the signals coming out of it is different to the outside region. Here there is no difference. In case of helium ion microscope, which we have seen this region before, that almost similar contrast to outside region, there is no changes because there is almost negligible interaction between the helium and the specimen. On the other hand, what we can see here again, it is a isolated graphite contrast for nitrogen ion. Nitrogen ion is significantly different to the helium ion microscope. This portion is SiO2. SiO2 almost have similar contrast as that of graphite, isolated graphite, but here we can see that SiO2 contrast and graphite contrast is significantly different. Same is not observed with a helium ion microscope. So, this indicates that by changing the ion, in some cases we could see a drastic difference in the contrast or the ion induced secondary electron yield, and thereby helping us to distinguish the presence of different specimen in the material. This is the effect of ions on the contrast mechanism, which is not possible in scanning electron microscope. Then if we go to imaging mode, we have a couple of imaging modes, such as let us say high resolution imaging mode. In case of high resolution imaging mode, we should always use the secondary electrons, that is SE1, because ion induced secondary electrons or ISI1, they mostly emerge from a smallest area of the specimen, and therefore, as they emerge from the smallest area, because their interaction volume is also small, because the higher mass of the ions as compared to the electrons, and the signal or nothing but the secondary electrons generated because of the ions comes from a smallest area, they give the best resolution in the scanning microscopic technique. Certainly, if we want high resolution imaging, we can prefer to use higher primary energy, that is smaller wavelength. Smaller wavelength means we can use smaller spot size. Smaller spot size means we can have a better resolution. Third point is a low beam current. Low beam current is important because it provides enough signal unless and until it provides enough signal to noise ratio. A low beam current means there will be less surface damage. First thing, there will be less surface damage and there will be less charging in case the sample is insulating in nature, and thereby it can give the best resolution. As per the studies, the best result is obtained with a beam current less than 0.2 picoampere in a Zeiss Orion helium ion microscope. So, here you see a large depth of field imaging. Image with a large depth of field is an advantage with the helium ion microscope. A large depth of field can be achieved if we can use a small angle of aperture, that is alpha can be need to be very small. But if we go for helium ion microscope, if we go for scanning electron microscope, we cannot make this alpha to be very small. If we make alpha to be very small, then there will be a significant increase in the diffraction aberration, and that diffraction aberration will lead to produce no sharp image, thereby the alpha value has to be an optimum. Alpha value cannot be made too small because diffraction aberration is inversely proportional to the angle of aperture. But on the other hand, in case of helium ion microscope, we have a liberty to use small angle aperture because in this case the alpha that means alpha can be made smaller because lambda or wavelength of the ions is much smaller. As the wavelength of the ions are much smaller, diffraction is much less, and thereby we have it can allow us to use a smaller angle of aperture, that is alpha, and with a smaller angle of aperture we can have large depth of field imaging. As you see in this image, depth of field is nothing but the range of position of the object at which a focused image can be collected. As you see, the bottom part of this region are well focused as that of the top part. So, for example, sample have a feature something like that. We can take a image with both bottom part and top part are well focused. If both are well focused, so the range, this range is called depth of field. This is much larger in case of helium ion microscope as compared to the scanning electron microscope, and certainly much higher than optical microscope. So, this is another advantage of helium ion microscope, where we can collect images with a large depth of field. Imaging the insulating sample. Imaging the insulating sample is another important requirement in the microscopic technique, because as we have seen in case of scanning electron microscope, charging occurs when electrons accumulate at the surface, and this charging electrons will accumulate when the sample is insulating in nature, because electrons cannot travel inside the specimen and cannot go to the ground. So, therefore, electrons will be accumulated in the surface, and that leads to distortion of the image, and the image we can see the bright line, dark line, and also the image can shift. Some region will be very bright, some region will be dark. Such kind of distortion occurs when we take the image for an insulating sample. But on the other hand, in case of helium ion microscope or ion microscope, this charging effect can be efficiently controlled. Here we can use a much smaller beam current. You may ask, then we can also use much smaller beam current in scanning electron microscope. Actually, if you use much smaller beam current in a scanning electron microscope, you will not get enough signal, because the yield of secondary electrons is much lower in a electron microscope as compared to the helium ion microscope. In case of ion microscope, let us say helium ion microscope, one ion bombarding on the specimen will produce 3 to 8 number of secondary electrons. One ions will produce 3 to 8 secondary electrons. On the other hand, one electron in scanning electron microscope will produce only 1 to 1.5 number of secondary electrons. So, the yield of secondary electrons is much higher in helium ion microscope. Therefore, we can use a smaller beam current, and then we can get enough number of secondary electrons or signal to create an image. Therefore, in a helium ion microscope we can use smaller beam current. When a smaller beam current is used, less number of such electrons or ions to be travel inside the specimen, and that would not cause the charging. Second possibility is the use of backscattered helium ions for the imaging purpose. Instead of using secondary electrons for the imaging purpose, we can use backscattered ions for the imaging purpose, and the backscattered ions have a quite large energy, quite high energy of around 10 keV, and that high energy will not be affected by the charging occurred on the surface of a insulating specimen. In the scanning electron microscope, we have also possibility to use backscattered electrons for the imaging purpose, but backscattered electrons have an energy greater than 50 electron volts, much lower than the energy of the backscattered ions. As their energy is much lower than the backscattered ions, they are affected by the voltage created because of charge accumulation. Therefore, backscattered electron image also get influenced by the charging, surface charging, though relatively less as compared to the secondary electron image from the SEM. And the third way to image the insulating sample is to use an electron flood gun, because when ions are positively charged, when such charges are accumulated on the specimen, then we can use a electron flood gun to neutralize the charge, surface positive charge, and that way we can image the insulating sample. Here you see in the right side a backscattered ion image of a pattern chromium on the quartz. Quartz is, this is the quartz SiO2, and this is what the chromium. First of all, what you can notice here, that there is a significant contrast difference between the chromium and SiO2. So, it is nothing but providing information about the specimen that certain region have one material, this region have one material and another region have another material because of this brightness difference. Here more number of backscattered ions comes out and they have a much higher energy as compared to here what is coming from SiO2, and that also gives us information about the atomic number of this specimen to an extent. If we use scanning electron microscope to image this kind of sample, it is almost impossible, because SiO2 is highly insulating sample and it will completely a very strong charging will occur on the surface, will not provide you a clear or distinct image of the specimen. On the other hand, what we see here very clear image of an insulating sample using backscattered ions. We can also advantage of doing biological sample using helium ion microscope. Normally biological samples are studied by 3 microscopic technique. One is fluorescence microscopic technique and other is electron microscopic technique. In case of fluorescence microscopic technique, we use those are labeled, those biological sample are labeled, and when the imaging occur, we see the position where those are labeled, the position which are labeled, not the overall morphology of the specimen using the fluorescence microscopy. And when the biomaterial labeling is done with a biometric material, that means it has more number of sample preparation steps, rather it is not straightforward or direct. On the other hand, in case of electron microscope, in case of transmission electron microscope, let us say the sample has to be cut to thin sections, so that the electrons should pass through the specimen and produce the image, and it would give you 2D image. It will give you 2D image for the specimen, and again the specimen preparation technique is not that straightforward or easy. One can do scanning electron microscope, it can certainly give 3D image, but most of the biological sample are insulating in nature. So, as they are insulating in nature, that sample must be coated with a metal, conducting metal, to avoid charging to be done. So, as a metal coating is provided on the specimen of the biological sample, it would not provide the accurate or precise surface morphology of the sample, because as we can see here, I have one example what the difference between when metal coating is made or when metal coating is not made. So, here is a biological sample, it is a stem cell sample prepared by critical point dryer system. In the top one is coated with a gold, it is a stem cell, and in the middle there is a almost like a spherical structure that is a type of called neurons type of things, that is round and spherical, from where we have like many lamellar type of structures are coming. These lamellar structures are arising. There is a filamentous type of structures coming out. This is a low magnification image, and this is top one is a SEM image. These are all SEM image. but on the other hand these are the helium ion microscopic image which is not coated. These are HIM image, helium ion microscopy image, this is SEM image. This is coated with a gold metal, and if we look at these two images, certainly it appears here that more 3D features with the surface having more clarity compared to here. On the other hand, if we look at here, we can see the difference of the surface of the cell. Here it is appearing as a particle morphology. On the other hand, it is appearing here as a pores here, as you see these are pores type of structure. The correct morphology is this, not this. Here these particles are nothing but the gold particle. When gold is coated, what we see actually not the surface of the cell membrane or cell, but rather the metal coating provided on the specimen. This is what happened for when a metal coating is done to a biological sample. On the other hand, we can see clear surface image of the using the helium ion microscope without any distortion or any other features, or the correct features we are seeing in the helium ion microscope. This is the advantage of helium ion microscope for studying the biological sample, therefore can be more trusted. Another example here, some biological sample. Here is a predator worms. The image reveals the high depth of field. In the left side, as you see, it is a high depth of field image, and in the B1 it was A1 is a native sample, B was this milling by the neon ions revealing the primary tooth here. The surface, this portion is milled away by using the neon ions, and we can see inside its tooth, and again the outer sheath was removed by milling by neon ions, and we could see the features, and then the D1 is the pattern line using a low fluence of neon ions. How we can create a features or break or damage a particular region by using the ions on this specimen. This is the advantage of helium ion microscope as compared to the scanning electron microscope. So, in conclusion, what we have seen that topographical contrast is maximum with the ions as compared to the electrons as the source. So, the first important information, and ISI, the ion induced secondary electrons from helium ion microscope even provide material and composition contrast, which is not provided by the secondary electrons induced by the electron beam. Like backscattered electron, backscattered ion also give composition contrast, as we have seen just now the chromium and quartz difference contrast, as the energy of the backscattered ions depend on the target material. So, the target material atomic number changes, the yield of the backscattered electrons, the energy of the backscattered electrons will also change. And helium ion microscope provide also different contrast such as voltage contrast, magnetic contrast as that found in the SEM. All type of different contrast we found in SEM can also be seen in the helium ion microscope. Helium ion microscope provides higher surface resolution because of negligible contribution of the SE2 which comes out little away from the position where the incident beam strikes to the specimen. Charging effect can certainly be easily mitigated with helium ion microscope without a metal coating on the specimen. For scanning electron microscope, we do need to coat a metal layer, conducting layer to avoid the charging, but here without metal coating given, we can do the imaging and see the clear surface. Helium ion microscope provides a much higher resolution, contrast and more detailed information in comparison to standard electron microscopic methods used in biology and medicine. So, all these information, all these points that you have seen here where helium ion microscope is much superior as compared to the scanning electron microscope. More details you can find from these books. These are the references, and thank you very much.